First of all, hello to everyone. The title of our project is Placental microRNAs as potential regulators in tumor processes. This project was a part of my undergraduate thesis as a biologist at the Pontificia Universidad Javeriana, Bogotá. The aim of our project was to identify those microRNAs that are highly expressed in placenta compared to cancerous tissues, as well to recognize through functional enrichment the metabolic pathways regulated by these placental microRNAs. This because we wanted to consider the similarities between trophoblasts, which are a type of cell really key to the beginning of a pregnancy and to embryo implantation, and tumoral cells, because they share some biological processes that are recognized as hallmarks of cancers, such as angiogenesis, proliferation, evasion of immune control, resistance of apoptosis, among other hallmarks of cancer. On the other hand, we wanted to consider microRNAs because these non-coding RNAs are key to regulatory control in many biological processes because they target messenger RNA and they have also an important role as biomarkers to identify different types of cancer in precision medicine. This is why we wanted to use microRNAs also because there are 2,500 microRNAs in the human body. 700 of these microRNAs are expressed in placenta and some are specific of placenta, like the microRNAs that confirm the cluster of the chromosome 19. So in order to accomplish this, we obtained data of raw reads of microRNAs for placenta and for healthy and tumor tissues. These tissues were breast, cervix, uterus, and prostate. The databases we used were the GEO, the Gene Expression Omnibus, and all the data that were available from the TCGA project in the GDC, the Genomic Data Commons, for cancerous tissues. We performed on these raw reads a differential expression analysis after normalization of the data between placenta and healthy tissues and also between placenta and cancerous tissues using R and bioconductor packages such as Lima, Etcher, and Glima. And when we found those microRNAs that were highly expressed in placental tissue, we performed functional annotation of these differential expressed microRNAs using Diana-Mir path to do an enrichment of the KET pathways. After performing the differential expression analysis, we found 78 microRNAs in placental tissue that are highly regulated and highly expressed and are absent in cancerous tissues. Some of these microRNAs were specific to placenta, included the microRNAs of the cluster of the chromosome 19. We found 45 of the microRNAs that compose this cluster expressed in our placental samples. And when we performed a functional arrangement, we found that the signaling pathway that was mostly regulated by our placental microRNAs was the MAPK pathway. This is the reason why we wanted to focus on these genes that are a part of this pathway and are being regulated by placental microRNAs. And after this analysis, we could conclude that cell proliferation, angiogenesis, migration, and invasion are some processes that are highly regulated by placental microRNAs and are absent in cancer progression, and this is a reason why these microRNAs are important to look forward in order to propose them as markers or potential treatment in cancer therapy.